Great. Okay, thanks, Hazel. And today our session this afternoon will be on microfauna and mutualisms. And it's a good and well-packed session with a lot of diverse topics that we'll be talking about. Just to go over what the main topics include, the Pacific Northwest ecosystems are knit together with players of all sizes. This session will explore the roles of microbes, pollinators, and other small but mighty actors in the large stages of forests, rivers, and mountain lakes. How do changes like rising temperatures or the introduction of non-native species ripple through invertebrate communities and into the broader ecosystem? And so we'll be talking about that, and I welcome people to add your questions into the Q&A component at the bottom of the screen in Teams. And we'll be starting off our presentations with Dr. Erica Sarro Gustilo, who's a postdoctoral scholar at the University of Washington. And she'll be talking about some of my favorite wildlife species, bumblebees, specifically what's driving range contractions for these fabulous creatures. Thank you.
Awesome. Thanks, John. Everybody can see my screen in full screen and myself.
Yes, it looks good.
Awesome. We'll start my timer. All right, well, thanks for having me out here. I am a postdoc at the University of Washington in Barry Brossi's lab, and I'm going to talk to you today about a project that I have planned for this upcoming field season. I have not started it yet, but it's going to be looking into the mechanisms of range contractions in bumblebees. So over the past century or so, climate has been warming. This is a graph here showing climate global mean temperature over time, and you can see that there's an increase of about 1 degree Celsius in the past couple decades relative to a century ago, and this temperature increase is projected to continue increasing over the next decades and centuries. And in response to this warming temperature, organisms, in order to succeed, either need to adapt to this warming temperature or they need to move so that they remain in the areas of their kind of habitat suitability temperatures. And so what this can look like is you have like a theoretical species here that you find kind of at this latitudinal band that I have this red bar over a map of North America. As temperatures begin to warm, you would expect this species to basically shift northward in the northern hemisphere, where it disappears from the warmer edge of its range on the south, and it starts moving into new territory in the northern edge, where that used to be too cold, but is now warm enough for them to survive. And we see this in lots of animals and plants across the world. But when we're looking specifically at bumblebees, we are not by and large seeing this pattern of kind of shifting species ranges. Instead, what we're observing across the globe is range contractions, where these bees are disappearing from the southern edge of their range where it's getting too warm, but they're not pushing into northern areas where they should presumably be suitable habitat now. And I've got a couple of citations here. This has been documented in a lot of different species in North America, in Europe, and in Asia. So this is kind of a global phenomenon happening with lots of different bumblebee species. And so with my research, I'm trying to look into kind of what the mechanisms are driving these contractions and ranges. And when we talk about mechanisms, they ultimately occur at the level of the individual organism. And so specifically, I'm interested in how organismal traits in an individual scale up to influence species ranges. And so for the sake of time, I'm not going to go too deep into the methods here because I haven't actually carried out this fieldwork just yet. But I'm just going to talk about a little bit about the things that I have planned for the upcoming season. So I'm trying to look into 3 different traits, the first of which is thermal tolerance. So how cold and how warm can individual bumblebees function in, what temperatures. I'm looking at overwintering survival, so how successful are individuals at surviving overwintering in different temperatures in colder or warmer environments. These are the queens overwintering by themselves underground. So what is their survival rates under different temperature regimes? And then I'm also interested in looking at interannual dispersal. And so this is kind of how far an individual queen travels from the time that she's born until the time that she starts a nest. And I'm interested in this in part just to kind of ask this basic question of like, are they even capable of dispersing far enough to track changing temperatures over time? And I'm interested in these traits as they relate to both climate, like I mentioned, kind of how do these traits operate under warmer regimes and cooler regimes. And also I'm interested in this as they relate to body size. And the reason for that is in bumblebees, body size is related to the amount of food they eat. It's environmentally determined, so the more food they get, the larger bodies they have. And so as climate's shifting and plants are moving around as well, it's possible that floral resources, which vary across their range, might not be tracking climate as quickly as these bees might otherwise be. So basically, if there are not enough food resources, maybe at this northern edge, that potentially could limit how big they can get and potentially limit some of these other traits. So I'm interested in kind of understanding how these traits vary across body sizes. And the plan is to conduct this work at Mount Rainier National Park this summer. And the reason for that is we've got this really dramatic elevational gradients, which can be used as a proxy for latitude and kind of look at these kind of different climates and different temperatures along that gradient. And thanks for listening. And here's my email in case you have any questions. Happy to follow up on anything later on. Thanks.
Excellent. Thank you, Dr. Gustilo. And we'll be having questions at the end. So if you have any questions for Dr. Gustilo, please throw it in the Q&A function in Zoom. Thank you. So next up, we have Madeleine Strait, who is a graduate student at the University of Washington. The effects of subalpine tree encroachment on subalpine meadow plants and pollinators will be the topic. Also, some work related to Mount Rainier. So please take it away, Madeleine.
All right, well, thank you. Okay. Present. All right. Hi, everyone. My name is Madeleine Strait. I'm a graduate student in the biology department at the University of Washington. And today I'm going to tell you a little bit about a project that I've been working on at Mount Rainier, looking at the impacts of tree encroachment on meadow plants and insect pollinators. Briefly, I want to quickly acknowledge some of the people that have helped with this project, specifically Beth Fallon in the National Park Service. I couldn't have done this project without her advice and help. The entire Brosi lab and my mentor, Barry Brosi, as well as these 3 undergraduate students, Nina, Stella, and Giovanna, who really helped with site selection and data collection and really great in the field. And then, of course, my funding sources. So to start off, I'm going to briefly explain what tree encroachment is. Tree encroachment is this phenomenon that we're observing in many mountain landscapes. And this is because climate varies along an elevational gradient. So as you go upwards in elevation, we have colder temperatures, more precipitation, usually in the form of snow, whereas at lower elevations, it can be warmer and less snow. And in the Pacific Northwest, we're observing that there are warmer temperatures and reduced snowpacks. And so these areas that were in higher elevation, colder, more snow previously are becoming more similar to lower elevation climates. And because of that, trees are then able to move upwards and encroach or invade into these meadows that were previously dominated by these herbaceous plants. And we can see a lot of that actually occurring within the park. And so I've shown here on the left just a photo of looking from Paradise up at Mount Rainier. And you can see I've highlighted here some of these young patches of trees. And so I'm really thinking about these early stages of encroachment, young trees sort of less than a meter tall. And actually in 1996, Regina Rockfort and Peterson went out and they actually documented that tree encroachment had been occurring since the 1930s in these meadows across Mount Rainier. And so my goal was to really understand how this tree encroachment phenomenon is impacting both the plant in the meadow community as well as the insect pollinators and their interactions. And so to do this, I set up 4 sites within the larger Paradise area. And at each of those sites, I had an open meadow. So this was an area of meadow where there were no trees established, and then an encroached meadow. And so an area nearby that had early stages of encroachment. And so these are just young saplings growing in the meadow. And at each of sites, I then set up transects in the plots, and I visited the sites weekly for the month of August with the help of those undergraduate students. And we recorded the floral community. And so we counted and identified every flowering plant within our transects. We observed plant pollinator interactions and collected the insects we observed visiting those flowers. And then we also collected pollen from the insects that we caught in the field. And this will allow us to expand the data to figure out more of the plants that insects are visiting from those pollen grains. And so I am just digging into the preliminary results because the insects are currently here in the lab at UW, and we're identifying them to like a finer taxonomic level. But just generally, I wanted to show you all some of the things we found. And so in the open meadows compared to the encroached meadows, we actually observed a similar number of interactions. So each of these bars here is showing the number of interactions that we observed, and it's divided by a functional category of pollinator. And so you can see from this that in both the open and encroached meadows, we saw a lot of syrphid flies. So that would be like hover flies. That was probably the pollinator that we observed. And then we saw a bunch of bumblebees, which is here as bombus, and then a few other taxonomic groups. Although generally it seems like the relative abundances of these different pollinators is relatively similar between the different types of meadows. Then we also looked at which plants these pollinators were visiting. And so whenever we caught an insect, we recorded the plant that we observed it visiting. And from that, we can create this network of interactions. And so what I'm showing you here on the left is all the different plant species we recorded pollinators visiting. And then on the right here is all of the pollinator groups. And any line that's connecting a plant and a pollinator is an interaction that we observed. And what I've done here is in gray are all the interactions that we observed in both the open meadows and the encroached meadows, whereas the interactions shown in green here were ones we only observed in the open meadows, and the ones in brown are the ones we only observed in the encroached meadows. And so there were definitely some interactions that we only observed in each type of meadow. Specifically, there seems to be a bunch of different interactions that only occurred in the encroached and not in the control. And so we're excited to sort of tease this out and figure out more about what's happening here. Then lastly, with the pollen that I am collecting from those insects, I will be able to use this method called pollen DNA metabarcoding, where we basically extract the DNA from the pollen, do some sequencing on it, and we can then determine which plant species that pollen grain came from. And that allows us to create a history of interactions for that insect that we collected. And this will allow us to really build out information on which floral resources are important for different pollinator groups, and get a sense of whether or not there's shifts in those preferences and those interactions when the trees are encroaching in the meadow. And I just want to say thank you all, and I'm excited. I will be continuing this work next summer, so this upcoming summer, and sort of expanding the sampling to include June and July, and maybe visiting sites more often. And I'm excited to hear if you guys have any suggestions. I would love it as we dig through the microscope and the insects back in the lab. Thank you all.
Thank you very much, Madeleine. Very good and excellent topic. Very important to figure out what is going on with all those interactions. So next up, we have Zach Farrand, who's a graduate student at the University of Wisconsin-Madison. And Zach will be speaking to us about adaptive mechanisms of montane ground beetles. We're keeping with our upper elevation theme for the moment. Go ahead, Zach.
All right, thanks, John. Everything look good? Audio, video?
Yes, I can see it. Thank you.
Excellent. All right, so I will continue the theme of Mount Rainier with my work. So go ahead and advance here. So my beetles of interest here are a genus of carabid or ground beetles. There's over 400 species in this genus, which is quite a lot even for a beetle. And they are very cold associated. So in the United States, at least, they reside in montane regions. So they can be, like you see on the left, at high elevation alpine snowfields, or like you see on the right, at lower to mid elevation snow-fed streams. And Mount Rainier is a particular hotspot of diversity of Nebria, where there are 12 species distributed along an altitudinal gradient that you see in this chart, where each species kind of inhabits its own elevational zone as you move up the mountain. So species that reside in environments like this are very prone to disturbance from glacial cycles. So all through the Pleistocene, there have been kind of cyclical, roughly 100,000 year cycles of glacial interglacial cycles. And the chart that we have here shows mean annual temperature at Paradise at Mount Rainier going back 500,000 years. And the impact this might have on Nebria are that during the interglacial cycles, they can spread out along the mountain. You can have high elevation species and low elevation species on the same mountain like we have right now. In the glacial periods, they all get pushed into common glacial refugia, and in the successive interglacial periods that can spread back out. So this sort of cyclical mix isolate mix can really drive diversification of species and really contributes to why we have over 400 species in the genus today. So my research question here is adaptation required for high elevation species transitions? So I'm especially interested in this movement here as alpine species move back up the mountain during interglacial periods. My research approach, which is documented in this 2024 publication, there's a lot to this paper, but I'll focus on just kind of one element of it. We were looking at identifying signs of natural selection in a high elevation species on Mount Rainier. So my study design looked like this, where I had two species on Mount Rainier, where there's a high elevation species called Nebria van Dyke and a lower elevation species called Nebria piperi. And then we also have a species from the Sierra Nevadas, which was called Nebria riverside, which is also a high elevation species, which I use for comparison, which will make sense in a second. So we sequenced RNA, which is transcribed DNA on its way to becoming a protein, and we use these sequences to test for natural selection in protein coding genes. So my comparison here was taking kind of pairwise species comparisons, where on the left, we're looking for candidate genes that could be under selection from that high elevation versus low elevation comparison on Mount Rainier. And on the right, we have a comparison between the two high elevation species, where one's from Rainier and one is from California. And the reason that we did this is we wanted to exclude signs of selection in Nebria van Dyke that are not related to that difference in elevation. So we kind of excluded genes from this high elevation comparison. And finally, with this final candidate gene set, we did an analysis called a functional enrichment, which is kind of a fancy way of saying that we looked at the functionality of these genes and kind of compared it to baseline, kind of how common these functional categories across the genome to find kind of exceptionally rare functional categories to really pare down our list into a manageable gene set. So our final results were a list of 9 genes that could be under selection that were from that high elevation versus low elevation comparison. And I'll just point out that 5 of these genes out of these 9 were related to hypoxia in one way or another. And what's really interesting about this in a montane system is that as you go up in elevation, atmospheric pressure declines. So these high elevation species are likely dealing with this novel or at least different abiotic stressor than low elevation species. So as we go up in elevation, say from sea level, we can experience maybe it's harder to breathe, kind of a metabolic stress. Well, this can stress out insects as well. So we really seem to find some evidence for adaptation to this abiotic stress unique to the high elevation species. And the conservation implications here are that as climate change is warming the lower elevations, there's already evidence that Nebria are moving up the mountain in response to climate change. Sort of, if low elevation species are perhaps lacking some of these hypoxic adaptations, kind of will they have what it takes to persist in this kind of new, perhaps more stressful environment in some ways? So thanks for watching, everybody. There are my acknowledgements, and I will leave it at that.
Thanks, Terrific. I've never thought of beetles breathing and their requirements in that light. So that's really illuminating. So thanks very much for that presentation. And again, just a reminder to everyone to, if you have questions, please put them in the Q&A tool in the Zoom feature, and we'll have time for those hopefully at the end of this session here. Okay, next up we have Jordan von Eggers, who is a PhD candidate at the University of Wyoming, and will be speaking to us on plankton DNA from mountain lake sediments. So we're moving into the aquatic realm. So Jordan, I cannot hear you yet.
Oh, how about
Okay,
now?
there we go.
Okay,
Oh, good.
awesome. Okay, and then you can see my screen okay?
Absolutely.
Great. Okay,
Thanks.
so hi everyone. My name is Jordan von Eggers. I'm a PhD candidate at the University of Wyoming. My advisors are Dr. Brian Schumann and Dr. Amy Christ, and today I'm going to talk about how lake sediments can track the response of plankton to human-induced stressors over the last few hundred years. So mountain lakes, like this lake in Mount Rainier, Mount Rainier National Park in Washington, are generally cold, possess few nutrients, and therefore have low productivity, and they're also naturally fishless. And the plankton communities in these lakes are comprised of 3 trophic levels. So we have zooplankton at the top of the trophic pyramid, followed by heterotrophic protists and rotifers, so just smaller taxa that also consume other organisms, and phytoplankton or algae. And today we're rapidly transforming mountain lakes and their plankton communities. And 3 of these ways include, well, the first is warming lake temperatures due to climate change. And this can affect plankton communities at multiple trophic levels, here shown by this triangle showing these 3 trophic levels we talked about, with the deltas indicating change. We also have introduced trout into these naturally fishless lakes, and this can have top-down effects that cascade down from higher trophic levels to lower trophic levels. And finally, we're adding nutrients through atmospheric nitrogen deposition through industrialization and agriculture. And this can really increase our primary productivity, which then can filter out from lower trophic levels to higher trophic levels. So for my PhD, I'm using the DNA of plankton stored in lake sediments to track how these 3 human influences affect plankton biodiversity over the last few hundred years. And so I collected sediment cores from Washington, California, and Wyoming from lakes with and without fish introductions. And so here's a map of all the lakes I cored, and the dates next to the lake names are when the lakes were first stocked with fish. And so in Washington, I collected 2 cores from Mount Rainier and 1 from North Cascades National Park. And so in total, I collected 6 cores from 6 fishless lakes and 8 lakes with fish introductions. And I chose these sites because it captures this gradient of temperature changes and also nitrogen deposition across the Western United States. And so here's some of the data. So on the top panel, I'm showing air temperature data as our measure of climate change. And here we see that Washington has the smallest degree of warming compared to Wyoming and California. And then on this bottom panel, I'm showing a proxy for atmospheric nitrogen deposition, which I measured in the sediment cores. And we see again that Washington's experienced the least amount of nitrogen deposition compared to Wyoming and California. And so from this, we may expect that plankton communities in Washington may be changing less than they are in Wyoming and California. And so that's actually what we ended up seeing. So here I'm assessing how much change we saw in the communities according to the DNA that's stored in the lake sediments, and how this is related to the amount of nitrogen deposition we saw in these 3 regions. And so this is also broken up by those 3 trophic levels we talked about. And essentially we see that for phytoplankton and zooplankton, the yellow and orange color representing Washington really has the smallest amount of community changes, shown by the low levels on that y-axis. And this also corresponds to the lowest amount of nitrogen deposition on the x-axis. But as you see in the middle here with the heterotrophic protists, this doesn't always hold true. So it appears that this group is not necessarily strongly affected by nitrogen deposition overall. And we also see similar outcomes with temperature as well. And this makes sense because both air temperature and nitrogen deposition are lowest in Washington. And so essentially it kind of appears that lakes in Washington may be slightly buffered in terms of how these human influences are affecting plankton communities overall compared to Washington and California, where we tend to see more rapid and kind of extreme changes in the community. And to illustrate this with one phytoplankton group, here I'm just showing the relative abundance of chlorophyte DNA over time from left to right. And the thin lines represent individual lakes, and the thick lines represent averages across the states. And so what we see is that chlorophyte DNA is declining in this lake sediment across all the cores over time. And this really coincides with the onset of nitrogen deposition. And so we also here see that Washington kind of has minimal changes compared to more drastic declines in Wyoming and California. And in contrast, when we look at the DNA of a large copepod that's in the zooplankton group, we see that fish are really the most important for these changes we're observing. And this is because the fish consume the large zooplankton here. So in this situation, state doesn't necessarily matter because they're kind of declining across all states. after fish introduction. So just to sum this up, we found fish introductions were really important for zooplankton communities. I didn't have time to talk about some of the heterotrophic protists and rotifers, but it seems like they're affected by fish and nitrogen potentially. And phytoplankton is really affected by lake temperatures and nitrogen deposition. And with that, I'll say thank you to my funding sources.
Terrific, Jordan. Thanks very much. That's great to see the comparisons with the different regions. And so we're going to have our next presentation taking a closer look at our corner of the country with Ty Stevenson, a graduate student at Western Washington University, talking about mountain lake benthic macroinvertebrate communities at Norca and North Cascades and Mount Rainier. So take it away, Ty. I can
Thank you,
see your
Sam.
slides.
Awesome.
Okay, great. I can hear you now.
Great. So my name is Ty Stevenson, and I'm a grad student at Western Washington University. And today I'll be talking about mountain lake benthic macroinvertebrate community change in the NCCN. So first, just want to talk about benthic macroinvertebrates. These are aquatic invertebrates that are large enough to be seen with the naked eye, and they live in or on the bottom of aquatic environments. These include insects, arachnids, worms, mollusks, amphipods, and other invertebrates, but usually where the bulk of them are insects, like this Lednia stonefly. And why do we care about benthic macroinvertebrates? Well, they're very diverse, and so they're good indicators of water quality and also environmental change in our aquatic ecosystems. They're also an important part of the food web, being prey items for a large number of organisms above them. And of course, they have intrinsic value. We care about them because they exist and we don't want them to go extinct. So benthic macroinvertebrate sampling is a part of NCCN's long-term aquatic monitoring program in 12 mountain lakes in Mount Rainier and North Cascades National Parks. And during this time period when sampling was occurring, 20 additional lakes had consecutive years of sampling that the parks did for other projects. So like Jordan said, most people, when they think about mountain lakes, they think about fish, and these fish, these lakes were historically fishless. They're cut off from fish populations, and so a unique community dominated by amphibians and macroinvertebrates develops in them. And we have, as a society, have stocked a lot of fish in these mountain lakes and found that it affects amphibian communities as well as macroinvertebrate communities. And due to this, many agencies have started, or over the last few decades, have removed fish purposely from some of these mountain lakes, including in the NCCN. So with this long-term data set, this gives me a unique opportunity to look at how these communities change over time. And we know that every year there's local extinctions and colonization of species in these lakes. So these lakes are changing their community every year. And so our question is, what are the typical colonization and extinction rates in the North Coast and Cascade Network lakes? And do fish presence or removal affect these rates of turnover? And understanding this is important for monitoring to understand what a normal rate of turnover looks like. So I want to talk about how we measure turnover. And so in this graphic, I have a year one community of a lake and a year two community. And as you can see, after year one species goes extinct from the lake and two species colonize the lake. And to calculate turnover, we would take the number of unique species, the numbers of species that occur in only one of the years of sampling, and divide it by the total number of species. And we would get, for this lake, we would get a value of 0.5. And this is a pretty high rate of change. As you can see, you're losing 5% of your species and then gaining 40% more the next year. So what do we see in the NCCN lakes? Over this time period from 2008 to 2022, across all lakes, we averaged a relatively high rate of turnover. In lakes with fish absent and lakes with fish present, the turnover averaged about 0.5, and there's a slightly higher rate of turnover in lakes where fish are removed during that time frame. So removing fish may contribute to higher turnover. And the next steps for this project are to further investigate what factors are influencing colonization and extinctions of species in these lakes outside of fish presence, such as ice cover duration in the winter and watershed vegetation. And I'd just like to thank all the National Park staff, biologists who have helped me with this project so far, and all the National Park technicians from 2008 and 2022 who helped collect all this lake data, including my advisor, Angela Strecker. Thank you.
Great, thanks very much, Ty. Very good. If anybody has questions for Ty, the other presenters so far, please put them in the Q&A function in Zoom. So we'll next move on to our next presenter, who is Naomi Wurtzel, an undergraduate student at the University of Puget Sound, who will be continuing our mountain lake discussions, especially about fish, the topic being using stable isotopes to study the influence of fish on mountain lake food webs. So Naomi, if you could start up your presentation. Okay, great, I can see it.
Awesome.
Okay, thank you.
All right. Hello, everybody. My name is Naomi Wurtzel, and I'm a junior at the University of Puget Sound. And last summer, I did a research internship with the Aquatic Ecology Department of Mount Rainier National Park. I'm going to be presenting on my research titled Influence of Fish on Aquatic Food Webs of Alpine Lakes in Mount Rainier National Park, a Stable Isotope Approach. Tagging along to the yearly mountain lakes monitoring program that occurs at the park, I was able to obtain samples for this project. All of the lakes within the park are historically fishless, so when choosing which lakes to sample to see what the impact of non-native fish are on the ecosystems, I chose lakes of fish histories. One lake was chosen to represent current fish presence, and as a main comparison, another was chosen as a lake where there are no fish observed due to removal efforts. Within my project, I obtained samples of fish through fin clips where available, native salamander tails, native tadpole tails where available, macroinvertebrates, macrophytes, and benthic photosynthesizers, examples of which are seen here in the table. We're now going to look at an isoscape, which will put the stable isotope geochemistry into an ecology setting. Based on existing literature, we can develop a theoretical framework of carbon and nitrogen values that reveals energy and nutrient flow through a lake ecosystem. Nitrogen isotope values can generally increase with trophic position, and carbon isotope values vary among major groups of primary producers, such as algae and macrophytes, and can also increase with trophic position. Primary consumers, such as caddisflies and snails, we expect to have low trophic position and utilize specific types of primary producers. Secondary and tertiary consumers, such as stoneflies, mayflies, and dragonflies, have higher trophic positions and potentially integrate across carbon sources at the base of the food web. Finally, top consumers, such as fish and amphibians, occupy the highest trophic positions. This is the theoretical isoscape that we'll use for this presentation. So we're now going to see if we can find this in the Mora Lakes. Our first question was if stable isotope analysis could be used as a tool to record predictable ecological variations, such as food web interactions in these lakes. So this is the same carbon and nitrogen isoscape as the last slide, but now we're going to add the data from Deadwood Lake, which is a lake that has fish present. Variation in the Deadwood food web reflects trophic structure and interactions. Within this lake, there's a couple of things to point out. First, both salamanders and frog tadpoles were sampled, and the amphibians have non-overlapping isotope values, with frog tadpoles at lower trophic position than salamanders, and body size may explain this difference, with salamanders consuming larger or higher trophic level prey. Second, both species of fish, trout and stickleback, are top consumers in this lake. They're both very different sized fish, but appear to be consuming prey at a similar trophic position, which could include amphibians and predatory macroinvertebrates, such as dragonflies. To understand if there's a change in the trophic position of salamanders and or a food web reorganization without fish present, we can look at LP19, our presumed fishless lake. Overall, there is no clear change in the trophic position of salamanders between the fish and fishless lakes, and the food web structure is similar. It has only been around 2 years with no fish observed in LP19, and changes may take longer to detect with stable isotope analysis, so additional sampling is key in this lake. The modest increase that we do see in the carbon isotope values of salamanders hints at the potential use of different lake microhabitats in LP19 without fish predators present. As some food for thought, we did also sampled aquatic taxa at 3 other lakes. We obtained trout samples from Cliff Lake and LH18, and there are some interesting similarities and differences in isotope values that hint at the role of lake-specific food web structure. For example, the rainbow are sitting lower nitrogen-wise compared to the rainbow trout that we saw in Deadwood. Additionally, with more samples, Lake Allen may provide an insight into a lake with a nearly absent fish population as an intermediate between Deadwood and LP19. We aim to sample all 5 lakes again next summer to investigate potential interannual change and to generate more robust data sets. I'd like to thank the entire Mora Aquatics team for their help with this project, and thank you for listening.
Great, thank you very much, Naomi. That was terrific. It's great to see the theoretical framing of a hypothesis like that for the trophic position versus primary production. So thanks for that. Please, everybody, if you have any questions for our group, add them into the Q&A feature. And we're going to move on to Russel Barsh, who is the director of the Center for the Historical Ecology of the Salish Sea. Will be talking to us on a different class of invertebrates, ticks and tick hosts in the San Juan Islands at low elevations. So take it away, Russell.
Yes, thank you. Well, we are a nonprofit conservation lab that works in the San Juan Islands. We are involved. Whoops. Can you see that? I've just got a.
I could see your slideshow before, and now it's back on the.
Oh, for heaven's sake.
No worries.
Yeah, all
Just
right.
take a moment.
We'll get
We
it
have
back.
time.
I got a message
There
from
we are.
Zoom. Okay,
Great.
So we're in the San Juan Islands. We work with the San Juan Islands National Historical Park and with the San Juan Islands National Monument, as well as many public and private landowners. Very different terrain than most presenters have been talking about. We are basically a biogeography lab. We're interested in archipelagos and the way in which, over time, communities disperse and change in an archipelago. And we do both historical work with, for example, peat cores and dendro, and working with contemporary populations of plants and animals like the sharp-tailed snake in this slide. The islands are a big tourist attraction, and the conventional wisdom for many years was that we didn't have any ticks. That one of the good things about visiting the islands was you wouldn't get bitten by a tick and you wouldn't get Lyme disease. Well, that was the conventional wisdom. Doctors and veterinarians told us a few years ago that they were confident that the islands had not yet been invaded by ticks. But it turns out not. We carried out, starting 3 years ago, a community effort inviting islanders to look on themselves and their pets for ticks, to send us the specimens that they found. And in 3 years, we've accumulated over 600 specimens from 6 of the islands. We also did some work on our own to collect ticks directly out of the environment using dragging, which is a standard method for environmental tick collection, and trapping small animals, small mammals in particular, and combing them for ticks. That wasn't very successful, actually, and you'll see in about 2 minutes why that was. So briefly, some results. First of all, the ticks in the islands go questing. They go looking for blood meals earlier than ticks on the mainland. And they are, in fact, at their peak right about now, typically in February and March. And this is an interesting phenomenon that is associated with climate change. We're seeing some evidence just over this 3 years that we've collected ticks of an earlier and earlier emergence and questing. So as the overall climate in the region warms, we are also seeing ticks here in the islands questing earlier. Most of our samples came from people and dogs. There's some sample bias in this, and dogs are excellent tick magnets with lots of hair or fur, but in fact, we are concerned that dogs are a critical part of the ecology of ticks here and the vector that introduced ticks and tick-borne diseases in recent years to the islands, particularly on public lands where people come visiting and they bring their dogs and walk their dogs, and frequently they come from the mainland. It's important to note that ticks can remain attached, feasting on a dog for up to 100 hours. So somebody could literally drive from the East Coast to the West Coast with a dog that has a tick on it and have it drop off here in the islands. So the dog part of the equation is introduction and dispersal. And very interesting part of this is the role that public lands managers can play in convincing people to check their dogs both before and after they go on public lands and to be more tick conscious. Because of this also, we find that ticks are about equally likely to be picked up in a residential area in the islands as they are in wildlands, where people are more likely to be thinking about protecting themselves from ticks. And does it matter? Well, about 10% of the ticks that we were able to test by qPCR screening were carrying a pathogenic um, bacterium or microparasite. Although the interest generally nowadays is in Borrelia, which is the organism involved in Lyme disease, what we're finding here in the islands is that the dominant pathogen or dominant disease vector potentially is Babesia, the organism that produces babesiosis. It's a Lyme-like disease produced by a Borrelia-like microparasite that infects red blood cells. And your likelihood of picking one up here is about 4 to 5% if you get bitten by a tick that actually is engorged with blood. Now, here's what's really interesting on this score. Babesiosis is basically a disease of the Northeast and the Midwest. There have been hardly any cases here in this area so far. And one of the reasons for that is that we appear to have novel species of Babesia here in the Northwest. That's a very interesting phylogenetic question. Where did they come from? And what adaptations have they undertaken traveling here from wherever and speciating? And how do they relate, for example, to Babesia duncani, which is found in California and has begun to be associated with actual symptoms in people? So we have a little bit of a phylogenetic and genomic question to follow up on that we're working with the Fred Hutchinson Cancer Institute on and working with their genomics people. Now, here's another interesting question, biogeography. Well, we find lots of ticks on two of the large inhabited islands, very few on Lopez Island, which is about 1/5 of the geography of the county and about 1/5 of its human population, but only about 4% of the ticks that people found on themselves and their pets. Why would that be? Well, the answer may be lizards. This has also been observed but not yet published in the Gulf Islands. But it does appear that alligator lizards are excellent hosts for tick nymphs. And we have found as many as 14 or 15 nymphs in the ears of an alligator lizard in the national park here. So the potential story here is that alligator lizards are the true endemic host, and that the distribution of alligator lizards determines the distribution of ticks. And sure enough, here's the distribution of alligator lizards from collections in the in the Global Biodiversity Information Facility. And you can see that they're in the northern part of San Juan County, the northern San Juan Islands, but not the southern ones, which includes Lopez, presumably because they arrived post-glacially by rafting down the Fraser River. And here's the distribution of ticks that we collected. So if I toggle between those, you can see that there's a strong correlation between the distribution of alligator lizards and the distribution of Ixodes pacificus. So we think that we are dealing with a combination of an endemic host, the alligator lizards, determining the basic distribution of ticks, and visitors with dogs introducing the microparasites, which perhaps have diversified and adapted here in the islands. And that's the question that we're following up on. Thank you.
Thank you, Russell. That was excellent. That's really amazing to think about the ticks in the Northwest and particularly on the San Juan Islands like that. So I appreciate your giving us that presentation. So any questions, please put them in the Q&A feature. And our last presenter is going to be Cody Spiegel, who is a graduate student at the University of California, San Diego. And Cody will be speaking to us about leaf litter and microbe-mediated decomposition in Olympic Peninsula rivers.
All right. How does that look?
I can see it. Great.
Perfect. All right. Hello, everyone. My name is Cody. I'm a PhD student in Dr. Sarah Jackwell's lab at UC San Diego. And today I'm going to part of my work that focuses on how leaf-associated microbes mediate decomposition in temperate rivers. And so this is more on the mechanisms underlying decomposition. So I first want to start by highlighting that ecosystems do not exist in isolation. Materials and organisms can be actively or passively exchanged across ecosystem boundaries. And so these two pictures show examples of important ecosystem processes like the release of nutrients from decomposing salmon carcasses that fertilize riparian soils, and terrestrial inputs like fallen leaves and trees that add carbon and nutrients into rivers. And so over time, ecosystems and the organisms within them can become dependent on these exchanges. And so our lab studies the exchange of riparian leaves into rivers. And as you can see in this picture from one of our field sites on the Olympic Peninsula, leaves are quite abundant and regulate decomposition and nutrient cycling. And so over the past decade, research in our lab has consistently found that when leaves from the surrounding trees into rivers, they decompose faster compared to if those leaves were placed into different rivers. And so this figure shows leaf mass remaining on the y-axis and the deployment river on the x-axis. And then the box plots on the far right show that leaves decomposed more when they were placed in their home rivers compared to when leaves were placed in more distant or away rivers. And so this phenomenon is referred to as the home field advantage. And so with faster decomposition, there's a greater release of carbon and nutrients. And although the home field advantage has been observed globally across different ecosystems, there's really no consensus on the underlying mechanisms. And so while many studies show that the quality and quantity of leaves entering rivers is an important driver of the home field advantage and decomposition more generally, another mechanism that is largely explored is the role of leaf-associated microbes. And so this diagram shows a leaf with microbes living on its surface and in the intercellular spaces. And so these leaf-associated microbes possess traits that may facilitate the decomposition process once leaves are submerged in water. And so to illustrate this a bit more, following this diagram from left to right, we see that leaf microbes in blue are transported into the river and mix with microbes from the water, which are shown in yellow. And then the interactions that occur when these two communities mix might be another mechanism responsible for the home field advantage. And so with that, we asked, how does the presence of leaf microbes influence decomposition? How does the presence of leaf microbes influence plant compound metabolization, which explores decomposition at a finer scale through the breakdown of compounds in the leaf? And then we hypothesized that the presence of leaf microbes drives the home field advantage pattern because their adaptive traits initiate decomposition and perhaps facilitate how microbes from the water colonize the leaf. And so to test this, we conducted a reciprocal leaf transplant experiment on the Hoko and Siku rivers with leaf-associated microbe presence and absence, which is illustrated in the diagram on the left. And then for the sake of time, I'm going to skip over the methods, but I'm happy to answer questions later. And then the photo on the right shows our leaf packs, which were secured to the riverbeds and left to decompose for 19 days. And so to answer our first question about leaf microbe presence and decomposition, we use leaf mass loss to calculate the magnitude of decomposition. And so this figure shows the percent magnitude of decomposition at home when leaf microbes were present or absent. And so when leaf microbes were present, which is seen in darker gray, there were significantly greater decomposition magnitudes compared to when leaf microbes were absent. And so this indicates that the home field advantage is really only seen when leaf microbes are present. But again, this is just mass loss, so we can probe this a bit further to understand what might be occurring at the plant compound level. And so during that same field season, we conducted another experiment with those same leaf packs to test how microbes are actually metabolizing these compounds from the leaves they most often encounter, or what is known as their home metabolites. And so this figure shows the metabolization of home metabolites on the y-axis with either microbes that are present or absent across different days of decomposition. And then so plant compound metabolization significantly increased with leaf microbe presence compared to absence, especially at earlier days of decomposition. And so just to summarize here, the home field advantage is only seen when leaf microbes are present. There was greater metabolization of home metabolites when leaf microbes are present, especially at earlier days. And then together, this suggests that leaf microbes play a proactive role in the home field advantage and perhaps mediate plant compound metabolization. And so we can hopefully use this information to better understand how exchanges, even at the micro scale, can mediate broader ecosystem level processes like carbon and nutrient cycling. And so with that, there's a lot more being analyzed, but I want to thank my lab, collaborators, and funding sources, and you all for listening.
Excellent job, Cody. That's great work with the leaf packs. I hadn't thought of that before. And yeah, the home field advantage is one of those areas where the closer you look at certain aspects of nature, the more surprises it comes up with for you. So thanks for that excellent presentation. So at this point, I'd like to invite everyone to come back on to all the panelists to come on to camera, if you will. And we'll have our question and answer session. And please, those in the audience, if you have a question for any of the presenters at this time, go ahead and add it to the question and answer feature or in the chat if you're unable to find that. I do have a few questions that are queued up. The first one is on the bumblebee presentation. So basically, the question being curious why bumblebees as opposed to other bee species. So I'm taking that question as being related to why study bumblebees as opposed to some of the other bee species in this particular range study?
Yeah, that's a great question. Most bee species, like most invertebrates, really, we have really little information about kind of what their ranges really are. Especially when you compare to like things like birds and large mammals, we really have not a very good sense of where these individuals are geographically. Bumblebees are one group of insects that is relatively more studied, and so we do have a little bit more information on these patterns. And so it's very possible that these range contractions are happening in other bees as well, but they're just not as well documented. So I'm kind of moving forward with one species that I have a background in, and that also it has better documentation.
Great, thank you. And I have a second question here that's about the beetle presentation. So that would be Zach. And what altitudes are you talking about when you're differentiating between low elevation and high elevation beetle species?
Yes, that's a great question. Thanks for that. So I'd say in general, Nebria can go from sea level to over 5,000 meters, which would be out in the Himalayas. I've collected Nebria over 4,000 meters in Colorado, which is exceptionally high. But in this study in particular, at Mount Rainier, Nebria van Dyke, the higher elevation species, was only around 1,600 meters, while the lower elevation species was around 900 meters. And then in California and the Sierras, the high elevation species was around 3,300 meters. So it to see signs of hypoxia adaptation in higher elevation species at Rainier kind of raises some really interesting questions about kind of how the insect respiratory system is responding to even what we might consider kind of more subtle changes in elevation, since they can get much higher in other parts of the range. So, yeah, thanks for that.
Excellent. Okay, we have another question from Angela Strucker for Jordan. A very cool project. Have you considered the role of atmospheric phosphorus deposition in addition to nitrogen?
I have thought about this, but I think one of the problems with phosphorus is that, if I remember this right, that we'd have to use something like diatom inferred phosphorus levels to like reconstruct past deposition, because I think that phosphorus can escape through the like it's metabolized in the sediments, if I remember that right. And so nitrogen has preservation in the sediments, but definitely phosphorus would have a big effect too. That's a good point.
Great. And so I'm curious for any of the presenters who would like to chime in on this. Is there any single abiotic factor that has had a surprisingly large impact on the ecosystems that you're studying? Because we're seeing, yeah.
I can
Russell,
go.
were you saying? Okay, yeah, go ahead, Ty.
Sorry, this is maybe not super surprising, but something that may not that I learned over the course of this is that the diversity of macroinvertebrates in the lakes decreases with elevation, which makes sense when you think about it, but I didn't realize until I looked at the data. So as you go farther up on the mountain, there's less species that live in the lakes.
Yeah, I mean, there's part of it where we learn more about the system when we look at it more closely. And some of it is that things are a little bit surprising. So with Russell's presentation, I was particularly interested in the Babesia. And I wonder about, if you look at the disjunct distribution of Babesia in North America, it seems the question that I have is about the oak woodlands, the role that oak woodland communities and that type of vegetation community might have a bearing on the types of ticks, and then, of course, the tick-borne pathogens that are borne by those ticks. Do you have any thoughts on that?
Well, it is interesting that we also have a distribution of native oak woodlands islands that fits the tick distribution and the lizard distribution. But that may be more a result of the initial colonization, the pathway of colonization by rafting from the Fraser, and the way we have a kind of a north to south colonization of the islands in post-glacial times. Um, I think that what was originally considered here was that the oaks were the reason why we had some animals in the northern parts of the islands, right? But I think that it's not the cause. It's just one more effect of the same underlying mechanism.
Yeah.
At the same time, when you asked about abiotic factors, roads, paving, access, people with pets, very, very big introduction and distribution mechanism. And if we're talking about national parks, the national parks are all managing access. And one thing that we've had discussions with our national park colleagues about is the extent to which people should be aware that where they go, they may be doing things like dropping off infected ticks, and that the going off trail and bringing uninspected dogs and things like that are accelerating the process of dispersal of some very interesting and perhaps unpleasant organisms.
So I have another question for this one's for Cody with regard to the home field advantage, one of those mind-blowing things of how does that work? Um, so with regard to the role of leaf-associated microbes on that decomposition variance as you move away from the home river, as you're calling it, how much might that have to do with species associations between the microbe and the plant that is bearing the microbe?
It's a good question. A lot. So, plants are recruiting microbes from the soil, from seeds, and so I think over time, once the leaves go into the river, there's definitely a strong association between the microbes that are on the leaf, and then once they actually go into the river as well. And so I didn't really touch on it for this talk, but I'm looking more at the kind of gene expression level from those leaf samples to see what genes are regulating it. And then I'm also tracking the changes in the chemical profile of the leaves too. And so that's kind of going to hopefully integrate that and kind of get a better idea to see like how well are those leaf microbes matched to the, I guess, like suite of chemicals that are in the leaves. Yeah.
Excellent. Great. And so I have a question also for Naomi in particular, but I suppose it would also be for Ty and Jordan. It's about the role of fish on the tropic levels in lakes and how much time it would take to reset the tropic hierarchy in a lake after removing fish. Any thoughts on that, Naomi?
Yeah, we're really unsure right now. We're hoping that this project will continue for a number of years, with more Puget Sound students doing the same research internship that I was able to do last summer, just to increase that data set and have a not like a time scale data set. So right now we're unsure like what the time scale is for seeing that impact, but hopefully in coming years we'll be able to have a better idea of that with more data.
Thank you. Ty or Jordan, any thoughts on that?
Yeah,
Any addition?
just thinking about turnover, looking at some of the lakes that had the highest turnover, or most significant turnover between years, um, were lakes that had fish removed, and then it seemed to, like, I guess I kind of expected it to spike and then kind of settle out, but it seemed to remain pretty high. So, um, I mean, it makes sense if you remove a top predator that you're going to kind of shuffle everything around, and people or species are going to be finding their place and stuff. But um, yeah, I'll have to look into it further. But it seems like it remains at a higher level, which for at least just the time frame of this study, which 5 or 6 years or something. So that's interesting, I think.
Yeah. And it could be with if the lake is more isolated, it'll take more time for things to colonize in, although they
Sure.
move around. Okay. Jordan, any thoughts that you'd like to add? Not to put you on the spot.
No. This is really interesting because I was talking to Ashley Rawhauser from North Cascades National Park, and he was explaining that at least for zooplankton, they did I think a rotenone treatment in one of the lakes up there to remove fish, and then within like 5 years, some of the zooplankton were back. And so, at least from what I've read too, because they've done some fish removal in the Sierras as well, is that as long as you like haven't depleted the egg bank for zooplankton, they can kind of come back pretty quick. And so like the eggs are stored in the sediment and then can come back. But I know they've also seeded lakes too to help them kind of repopulate. But yeah, sounds like time scales can vary, but sounds like they can bounce back relatively fast.